Hello, I'm Yifeng. It's my great pleasure to introduce our work on the inference of transcriptional programs across cancers using interpretable deep learning methods. This is a joint work with Xiaojun, Drake, Russell, Xinhua, and Hatice. It is widely accepted that cancer is mostly caused by somatic mutations, copy number alterations, or epigenetic modifications that deregulate the signaling pathways. The signaling pathways will first send information to the nucleus, modulate activity of transcription factors, leading to up or downregulation of target gene expression, which will lead to the aberrant phenotype, for example, the cell proliferation. As you can see, the transcription factors are the main link between the upstream signaling pathways and downstream gene expression programs. Therefore, it is important to take into account the transcription factor when we study the signaling pathways. We believe the pan-cancer modeling of regulatory programs could be informative in our project for the following reasons. First of all, the extensive pan-cancer genomic analysis in the past have shown that the same genes and pathways are targeted by somatic alterations across multiple tumor types. For example, the gynecologic cancers such as uterine or ovarian cancers show the frequent TP53 somatic mutations as well as widespread somatic copy number alterations. So this might suggest that similar transcription factors may also be dysregulated across cancers. And secondly, the similarities between cancer types can also inform new therapies. Especially, this is very important for the rare cancers. And lastly, in our model, the extensive training data from more common tumor types can also compensate for smaller sample sizes in similar but rarer cancers. The relationships between the somatic alterations and the transcriptional factors are not linear, meaning that the effects of upstream alterations are not equal. For example, the driver mutations will more likely to have a larger impact compared to the passenger mutations. And also there exist complex interactions between the genes. For example, the mutual exclusivity, and finally, the role of genomic alterations is also context specific, depending on the biological environment. In order to model these nonlinear relationships, we introduce the attention mechanism. Attention mechanism is a deep learning method that has already been widely used in computer vision and natural language processing. It is a method that tries to assign the importance weights to all the input features, and these importance weights are calculated in a contextual manner, meaning that it depends on all the input features. Here I give two examples in image classification and machine reading separately. So in the example of image classification, we can see that the regions corresponding to the predicted labels are given by the model with highest weights, while in the example of machine reading, we can also see that only the words that are most important are given the weights, highest weights. Therefore, we can see that attention weights can provide the model some interpretability. Actually, in a lot of applications, it has been shown that attention mechanism can also improve the model performance. Our model takes the patient's somatic alterations at the input and utilizes ataxic data to define a prior for the cancer-specific regulatory network. The model will finally predict the patient's RNA-seq in a personalized way for all the 17 cancer types. Here are the model details. We name our model Citrus, which is an interpretable deep learning model. Citrus takes the somatic alterations as the input and then utilizes the self-attention mechanism to assign different weights for these inputs. That is alpha 1 to alpha m, so that a somatic alteration with a higher weight will have larger effects on the downstream process. After that is a fully connected layer, and we will get the transcription factor layer. This layer represents the inferred TF activities. Finally, using the ataxic prior, we can apply the sparse connections between the TF layer and the final output layer of expression. Also, in order to take into account the heterogeneity of different cancer types, we introduced additional cancer type input. As for data sources, we collected the ataxic of 400 tumors and both somatic alteration data as well as RNA-seq data of around 6,000 tumors in 17 cancer types, both from TCGA portal. We used tumor ataxic profiling data to determine the set of TFs that potentially regulate each gene. Briefly, we started with an atlas of chromatin-accessible events derived from tumors of the cancer types to be analyzed. We represented each gene by its feature vector of TF binding scores, where motif information was summarized across all promoter, intronic, and intergenic chromatin-accessible sites assigned to the gene. As sanity check, we first conducted a statistical evaluation on the gene expression prediction task. We compiled our deep learning method, Citrus, to a bilinear method called affinity regression on this task. And we can see that Citrus consistently outperforms affinity regression across all the cancer types in the sense of Spearman correlation. This indicates that Citrus has a better ability in modeling the nonlinear relationship between the somatic alterations and the gene expression. Then we take a closer look into the attention weights. Here we show both the attention weights and the frequency of each mutation in pan-cancer analysis, and we can see overall they have some correlations, especially for the top ones. For example, TP53 and PIK3CA. However, we can still see that for a lot of mutations, although they are frequently mutated, they are not assigned with a high weight, meaning that our model does not assume them to be as important as the mutations appear in the top position of the figure. We also show the results of attention weights in different cancers such as breast cancer and head and neck cancer. And we can see that for different cancer types, these sets of most important somatic alterations are also different. We next use features across tumor types to learn patient-specific TF activities. The clustering of tumor by inferred TF activities as derived from the model largely recovered the distinction between the major tumor types. In particular, samples with squamous morphology components are grouped together. These include the bladder, cervical, and endocervical. As for geo, head and neck, and lung squamous. And similarly, tumor with tissue or organ similarities or proximity are also grouped together. These include neuroendocrine and glioma tumors, clear cell and papillary renal carcinomas, a gastrointestinal group, and breast and endometrial cancer. We conducted k-means clustering on inferred TF activities for each cancer type to get the subtypes, and then conducted hierarchical clustering for both the cancer subtypes and TF activities. The TFs are in general grouped into 9 groups, which are enriched in different cancer types. We can observe major variations across different cancer types, and less but significant variations within each cancer type. These variations within a cancer type may be explained by the distinct mutation or copy number alteration profiles of different subgroups. One advantage of our method is that we can explore the effects of mutations directly by conducting a virtual knockout simulation. Given a tumor patient, we can remove specific mutation from the input of the citrus model and check whether any downstream TFs are increased or decreased after the knockout. This simulation controls everything to be the same, except the interested mutations, and it is different from the traditional t-test analysis. Using this strategy, we conducted a systematic knockout in silico simulations for different mutations in breast cancer to infer their impacts on the inferred TF activities. We can find that in breast cancer, the mutation of PIK3CA will lead to decreasing TF activities of HIF1 alpha. If we take a closer look into the PIK3CA mutation in breast cancer, we can see the inferred HIF1 alpha activities are much lower in the mutant compared to that in the wild type. Actually, previous studies have demonstrated that the PI3K-AKT signaling pathway regulates the expression of HIF1 pathway in hypoxial ischemia. Meanwhile, the HIF1 alpha RNA expression is less significant between the mutant and wild type. This indicates that TF activities may be a better metric compared to RNA expressions when we model the signaling pathways in cancer. Then, we focused on the activated TF activities caused by the TP53 mutation. We can find the regulation directions of TP53 mutation to the same TF are the same across different cancer types. We found the TP53 mutation significantly promotes the ETS2 TF activities in breast cancer. TP53 is a tumor suppressor gene that suffers from missense mutations. These missense mutations will express a mutant form of the p53 protein and enable the oncogenes such as the ETS2. Again, we can find the ETS2 RNA levels are much less different in TP53 mutants and wild-type samples, which is consistent with our previous observations for the PIK3CA mutation and HIF1 alpha expression in breast cancer. In summary, we proposed a model, Citrus, which is a deep learning model to simulate the transcriptional programs in pan-cancer. Citrus utilizes self-attention mechanism to capture the nonlinear effects of the somatic alterations and integrates the attack-seq as a knowledge base in the neural network. As for the next step, we hope to further explore the potential clinical relevance of our model and the translational applications. This work wouldn't be possible without our great collaborators, Hatice, Xinhua, Russell, Xiaojun, and Jake. Xiaojun is also the co-first author of this work. ISMB Glue Lab is looking for students and postdocs, so feel free to reach out if you have interest. Thank you.